examples PowerPoint will go to the unit test. Okay. Okay, so please ask any questions you have in there from everything, and then we'll take up the quiz and take up everything. And I also wanted to let you know there is, because when we started the course, we didn't know this, but now I know there is a parents' night. It's going to happen around October 25, and it's virtual, of course. And there is also going to be interim report cards the first week of October. I'm still having trouble wrapping my head around the difference between independent and dependent variables. Okay, so when we take out the quiz right now, we'll talk about it. But basically, one trick you have to follow is whenever you've chosen something, right? You start an experiment. The first thought you had was about the independent variable. Then you think about the dependent. Think about when you guys planned your experiments. You always thought first, what are you comparing? That's always you comes to mind first. Then you figure out, okay, I'm comparing these two chemicals. I want to see how they boil. I want to see how long they take to do this. I want to see, so it's always second. The dependent is always second. Just all the deadlines for everything are in your calendar in the Google Classroom, and you will be able to see that the enzymes project is due on October the 13th, the day after Thanksgiving. Okay, and this is a poster. I saw you returned yesterday's quiz with email, but try to open it and said you needed permission. Oh, well. Don't worry, we're going to, you won't be able to see the answers that are right or wrong, you'll just be able to see your mark. No worries, Lisa. So I'm going to take up now the quiz and you will be able to see all the answers that are right or wrong. Okay? But in TeachAssist, you'll be able to see your mark even though it doesn't count. Maybe you're wearing your school account because I could see it. Okay. Yeah, if you're not in your school account, you're going to do anything. Yeah. No worries. Okay. Who has any more questions, or we go and take up the quiz and then we can start learning about lipids? How about we go do that? Okay, so I'm going to present. This is giving me so much trouble. So I'm going to present. You should be able to see the window of what I'm opening, and I'm going to open our quiz. So in classwork, our quiz, and I'm going to open the quiz, and then we'll see the results. And the statistics. Let me know if you cannot see stuff. Okay, can you see that? Okay, perfect. So what you see here is that overall our results are pretty decent, okay? The median is 7 out of 10, the average is 7 out of 10, so there's a 70. That's a pretty high average, okay? Normally averages are about 60. So that means that most of you are not having trouble with this and you're doing okay. So, but there's some questions that give people trouble, so what I'm going to do is pause on those questions. So this question most people had right, but maybe you guys forgot that you can choose more than one option, because last time we had an issue with that and we couldn't do it, but I fixed it and you should be able to do it. So if you look in here, all the options are the right answer. You have to have all of them to have an experiment. So if you didn't choose all, it's probably more a glitch than anything else. And that means that everybody got their answer right. But everybody should have had the same number of all of them. Now in these questions, says an independent variable has to, and most people had this right, be selected by the experimenter. It has to be the one that you chose. But some people put these answers, and that means that there's a bit of confusion. So I'm going to pause for a second and explain this. An independent variable doesn't have to be numerical or quantitative, which is the same term. So it's funny that most people chose this. Chose this. This is it means that you can count it. And an example is when you compare water with ethanol to see which one has higher solubility. I think one of your team members is doing that, one of your classmates is doing that. This experiment you cannot count. You cannot say ethanol is 3 and water is 2. It's not quantitative, the independent. You're comparing two things. But it is an independent, and that is selected by you guys. So that is the only thing that defines an independent. And be the only variable obviously is wrong because you need a dependent. Okay? Does anybody have any questions about that one? Okay, I'll come back. Oh, so choosing water and ethanol becomes the independent. Yeah, whenever you're comparing two things, whatever you were comparing. Okay, I want to see if Tide or I don't know another brand washes my socks better. The brands are the independent, and the socks cleanliness is the dependent. So whatever you compare is the independent. So they don't have to be quantitative, okay? Or numerical. A dependent variable has to, and in these examples, a few people answer wrong. It doesn't always have to be quantitative. I prefer if it's quantitative in grade 12. When you're in grade 10, I let you do experiments that are not quantitative. But in grade 12, I'm more demanding, okay? But you can see simply if it's had an effect, if it changed the color, those are qualitative results. It cannot be the only variable used because you need the independent. And it has to respond to the independent. Well, for it to make sense, it does. Now, it could be that your results are null, it doesn't change, but it still has to be, the idea is it's a variable that responds, okay? Anybody has questions about that? When we have the unit test, can you specify, that's funny, Emily. Somebody else asked me the same question in grade 10 class. If I specify in a very given question, here you can choose many, then you are going to choose many in that one, and that's kind of giving you a hint. So the only thing I can do is specify at the beginning, remember, you are capable of choosing more than one answer in some of the questions. And then you have to decide if you know it, okay? And I am sorry, but this is only helping you for next year when in university you have now questions that are 8 options. You have to train yourself to be able to cope with that. Even though it's stressful and difficult, it is the only way to write tests in university. So, okay, the only way is to practice. That's why we have also every week multiple choice. Okay. In this question, everybody had it right, so no worries. In this question, almost everybody had it right. The following is an experiment, and the reason is you have to choose which one is a true experiment, okay? So your teacher inflates a balloon and shows how it tracks paper. It's a demonstration, but there's no comparison, so therefore it's not an experiment. 2 cows walk at a given distance while they eat. Well, unless the cows are deciding to test who can walk longer, there's no plan here for an experiment, so that doesn't work. Which one is a dependent variable? In this example, most people had it right, but there is a few people that just chose one of the 2 examples. Okay, but in the end, all of them work. So you have to say all the options work. They are all correct examples of dependent variables, right? The height of jumping when comparing, the empathy, so they're all options that are valid. Anybody has a question about that dependent question? That's a matching when you have, yeah. But in my quiz I only had circle options. That doesn't matter. It's a different kind of multiple choice. Doesn't matter if it's squares. Squares, it's true, you can have more than one match sometimes. Like when you have squares, it's for a true and false. But that doesn't mean that in multiple choice you're going to have multiple options. You can also have multiple options sometimes. Okay? Okay, but I was asking about this question of the dependent. Anybody has any question about that? Okay. In this question, a lot of people fell for this one. Look how few people had it right, 3 only. So let's walk through. Which one is the independent? the time when comparing the speed of two people. Why is that wrong? What's the independent in that experiment? How would you choose the time? I don't understand. You're comparing the speed of two people. What are you comparing? Guys, I'm not asking you what you would do in another circumstance. This description says the time when comparing the speed of two people. It doesn't say, oh, you went to the shop and walked a kilometer. The time when comparing the speed of two people. So what is it that I'm comparing? What is the independent? What am I comparing? The people. Who's faster? And what is the dependent? The speed. Thank you. The speed, not the time. So there's two variables. One is the two people. The other is the speed. Time is not even in the picture. Now, you're right. In practical terms, probably you have chosen here, you. Okay, Joseph, come, run 2 kilometers and I'll time you. And then I'll decide who's faster. But what I'm doing is dividing the distance of the 2 kilometers by the time, calculating the speed. I may have mechanically taken time. I may have done that, but what I calculated was the speed, and what I was measuring was the speed. So be careful, okay? Because if you take your time in that question, time is not even a variable in the experiment. No, time is not a control. It's just what occurs is that speed is a variable that contains distance and time. So we usually measure speed by fixing the distance and measure the time, but that's not the variable we're measuring. We're measuring speed. So if you are running track and field and your prof or your coach says, oh, your timing was so short, it's implied that you are faster because in the same distance you took less time. But if we're saying what we're comparing is the speed, then no, you are making the division between distance and time. You are right. But because the experiment is about speed, you cannot say the variable is time. You have to say the variable is speed. Okay? It's science. You have to be exact. If my goal is to measure the speed, then what I am ending up reporting is the speed. If my goal is measuring the time, then what I ended up reporting is the time. You have to be careful. But in any case, time is certainly, absolutely, and forever not the independent. So even if that was confusing, it is not the independent. The independent is the two people. No, the answer is the independent. The question is about the independent. So the answer here is not time or speed. It is the two people. Okay, so that even if you were confused by the question of time, time would not be the suitable answer anyways. Okay, here in this question, the acid when comparing vinegar of different concentrations to see their pH. So take a minute to think about it. Don't be impulsive, you get it all wrong. So the acid when comparing different concentrations of vinegar for their pH. Let me go back and read it again. The acid when comparing vinegar of different concentrations to see their pH. Why is that wrong? Why is the acid not the independent? Sorry, Ethan, I don't know if you're talking about the speed or the acid. The previous one, you're right. How they measure the time or all those details are not specified, but when they tell you comparing two people for their speed, it's clear the independent is the people and not the speed, okay? And time is just thrown in there to confuse you. Because you are comparing two vinegars, so vinegar could be the independent. Watch, let's come back and read. The acid when comparing vinegar of different concentrations to see their pH. So I'm comparing different vinegars, or what am I comparing? The concentration. I'm taking vinegars of different concentration and seeing if the pH is different. So my independent is concentration and my dependent is? Exactly.
I'm sorry, I have to ask this question because I'm not certain I understand for the previous one with the time. Sorry, one second. Sorry. So with the time one, the independent variable is something you choose, right?
Mm-hmm.
So, I mean, if you chose a time, right, for them to run, so let's say I'm going to measure your speed.
It doesn't say that.
No, that's the thing, right? Because I don't really know how the structure of the experiment would be, right? Because that could be an option based on what the information they gave you, right? Because you could still be comparing their speed if you gave them a time of 10 seconds, right? Because then you just measure also how far they went. Then you say, okay, well, this guy ran, 30 something feet per second. This guy ran, I don't know, 20 feet per second. So then you can still.
Wait. It's a very valid point. So let me say, I took Mary Jo and Michael and Meredith. I said, guys, I want to see who's faster. So you have to run 20 seconds and I'm going to see who goes further to compare your speed, right? Yeah, that's the experiment you're mentioning. And I said to them, I'm going to stop you in 20 seconds and to compare your speed. Is that the time the independent then? What is the time?
Well, I mean, you're comparing the speed, but if you chose the time to be 20 seconds.
If you chose the time and you told them all, this is your time, what is happening with time? What is time there?
It's a definite value, right? Well, I guess it's a constant because
it's a control. So it's not the independent. You're right, I chose the time. I may have said, hey, run 20 seconds. It's still not a variable because time is being fixed. So if I fixed it, it's, if anything, a control or a constant.
Okay, well then now I'm getting confused when we apply this to say the experiment that I'm actually doing, which is I thought the independent variable would be something
you chose.
Yeah, something I chose, right? So
they have to be two things.
Like the temperature, right? But if I chose the temperature to be negative 20 degrees Celsius, then it be a constant.
No, but it has to fulfill two conditions. It must be something you chose, but it must be a variable. If you have chosen to keep something constant, we cannot call that an independent variable. So it has to be both things, something that varies and I have chosen it. So if I say everybody gets this, then it doesn't vary. Everybody gets 2 minutes, then it doesn't vary, then it's not a variable. It has to be both conditions. I chose it and it varies. So you get this, you get this, you get this. Or I'm comparing you. So that's my variation, the individual that is doing the task. You see it then? It has to do both things. I chose it and it varies.
Okay, so then okay, because it's formative and I can ask questions about it. So my experiment is with the expending water when frozen, right? So we thought, I think you're cutting out. Am I cutting out?
Hello? Now I hear you.
Hello? You hear?
Yeah.
So how much of that did you hear? I'm pretty sure.
No, I heard nothing. Say it again.
Right. Okay. So with my experiment, because it's formative and I can ask questions about it, we thought the independent variable would be the temperature when comparing how much when looking at how much the water expands when freezing, right? We thought the independent variable would be the temperature, but if we chose a temperature of negative 20, then it's no longer a variable, right? It would be a constant.
Okay, chill, just a sec. You're right. If you only do one temperature, then it's a constant. But if you're comparing room temperature to the one freezing, you have two values. So then it is an independent variable.
Okay.
If you're comparing room temperature with frozen, then you have two values, then it's varying.
Okay.
Yeah? Does that help?
Well then, okay. So the independent variable for, just so I remembered for the question we were talking about in the first place, I can't even remember the options.
Oh, in the one that we were talking in the first place, we were talking about comparing two people for their speed. So in that example, you're comparing the people. You're saying this dude is way faster than this person. So your independent is varying because you have many different people who you want to see who's the fastest.
Okay.
Okay, and no worries. Concentration leads to pH, and Sunny answered pH therefore is the dependent, concentration is the independent. And Shanice fell in my trap, and she said, is the vinegar, which is kind of like what the question says, right? The question says, if we go here, the acid when comparing vinegar of different concentrations to see the pH. Saying the acid or saying the vinegar is not right. Why is that not right? The acid or the vinegar are not right. Concentration is right. Why saying acid or vinegar is not the right answer for the independent variable?
Wait, Miss.
Mm-hmm.
So can you tell us like what like basically an independent like a definition like what an independent variable is then? It's basically like.
Yeah, give me a second.
Yeah. No
I didn't even understand the acid one. Okay, so to go back, an independent variable is first of all a variable, so it must be something that varies. Okay, and it's called independent or manipulated. Both are equivalent names because it's a variable that you have selected to make your comparison. So in an experimental design, you always have something you want to compare. An experimental design requires something that you're comparing. So you must have something that varies and that you've selected because you're wanting to compare it. That is an independent variable or manipulated variable. A responding variable or dependent variable is the variable that will respond to the original one. So in that example, you have acid, which is in this case vinegar, of different concentrations. You want to see if that leads you to different pHs. That's your test. And so your independent is the concentration of the vinegar and your dependent is the pH that results. So wait a minute. And so in that example, when we say the acid or the vinegar, that is not a suitable answer for independent variable. Why is the acid or the vinegar not a suitable answer for the independent variable? Why is that not corresponding to my definition? What happens to saying the acid or the vinegar? Yeah, the acid or the vinegar is not a variable. Okay? The telephone is not a variable. It's a thing. Now, if I said the height of the telephone, that's a variable. My shoe is not a variable. The shoe size is a variable. Okay? A plate is not a variable. The mass of the plate is a variable. Okay? So we must fulfill two conditions. It must vary, and I have to select it, both. Otherwise, it's not an independent variable. So going back to Ethan's question, what if you choose to measure the speeds of different durations of... That's another question. That's another situation. It's not that one. That's a different question. That's not what I was going to do. I was going to compare the speed of different people, not compare the speed of different lengths of time. Yes, there's millions of other experiments I could do, but that's not that one. How could you define the constant? Any constant is anything that doesn't vary, that you kept the same. I think that if you were measuring someone's speed, seconds, okay, guys, we're wasting too much time on this. And you're getting kind of stuck on irrelevant aspects. That's a different experiment. Constant never changes and has no different stuff. Yeah, thank you, Olivia. But in general, enough to be in an experiment, it's the same. Anything that is constant is kept constant, is kept the same. It doesn't vary. Okay, I'm going to move on because otherwise we won't be able to learn about lipids. At what temperature does fungi, the population grow faster on bread? So in this example, almost everybody chose the right answer. This is a typical multiple choice question where the right answer, you know, is the right answer because it has more details. So whenever you have more details, it's a better design than the others that are more simplistic. So this is pretty straightforward. This one is a bit more tricky. Does water with sugar make a plant grow taller or than just water? So in this example, this experiment is decent. Compare the height of a plant grown with water with one grown with sugar. So this is a decent design. However, this has more stuff in it. Measure the height of a plant over time grown with water. So because it has more information, it's more detailed, it has more data, it's obviously a better design. So that's a better answer. Okay? Anybody has any question about that?
Wait a minute, so it's like kind of like the more detailed one, right? The more detailed one, which helps you give.
As long as it's not incorrect information.
Yeah.
But if it's giving you more information, more data, then that's a better design.
Okay.
Yeah? Now you're saying it's general enough to be that experiment. Speed of 10 seconds, see who's faster, 10 seconds, see who's faster. In that example, when you're doing that, so you got like, I want to see who's faster at 10 seconds. I want to see who's faster at 20 seconds. In that example, Ethan, you have two independents. The people is one independent, and the length of time is the second independent. That's a
possible to have independence?
Sorry?
Is it possible to have two independent variables?
You can have, yeah, when you do research in big, for instance, psychologists do research and they are studying many variables, how people behave. And these are complex things. They have sometimes 30.
Okay.
So you use very complex statistics to analyze the trends and all the curves and all the graphs. So yeah, you can have humongous.
Okay. Well, then I guess my point originally would be then that the question was maybe a little bit too ambiguous with the structure.
No, I disagree completely. Your experiment is cool and it has two variables, but it's not answering that experiment, because in that experiment, your goal was comparing the speed of two people.
Yeah.
Not comparing the speed of two people at different times, which is your experiment. So no, it's not ambiguous.
Okay.
It's different goals. You have to be, it's science. You have to be exact. If my goal is simply to compare who's faster, I cannot do other things. That's why your question, your hypothesis, and your experiment must be tight. Because if look, hang on, the next question is perfect for this. Look, the next question, the one of the worms. So this, can water make a worm live longer? And a lot of people answer this question wrong. And they also, it's the same thing. So compare the length of the worms is obviously incorrect. Compare the worms in water and outside water is too vague, it doesn't give me information. The given area doesn't work. But compare the age of worms with or without water. Doesn't answer my question. Can water make a worm live longer? Does that answer my question? What do I need to do to answer that question? I want to know if water makes a worm live longer. What experiment do I need? Because that one doesn't work. Please, Nicole, I am sorry. Raise one with water, one another without water. Is that enough, Terija? You can do better than that. That's not enough. Yeah, Jusra, you need to know how long they live, and can I do it with one worm? Yeah, exactly. You need a bunch, right? And you go, okay, look, I had 100 worms living with water, 100 worms living without water, and the ones that were living with water, every day I went to check how many I found dead. And after they did the experiment for a year, I found out that the average age of death of the worms in water is 30 days, but the average age of death of the worms in no water is 20 days. So I can say that on average it seems worms live longer 10 days in water. Do you understand? So that experiment tells you the age. Maybe none of them died. Then I didn't answer my question at all. I should make that a sentence. Lol. Anyway, I'm back. Sorry to interrupt. No worries, Nicole. So we were talking about the experiment of the worms that a lot of people got wrong. And I was saying that the problem with this answer, which a lot of people selected, is that it doesn't let the worms die. Compare the age of worms growing without water. It doesn't tell you if they died. And if they haven't died, I didn't answer the question at all. Okay? Because I haven't finished the experiment. You need to have a sample of worms living with water and a sample with worms living without water, seeing how old they are when they die. Then you can say, okay. Okay, we're cool? But no, not only that, multiple worms, you need to let them die. Otherwise, you didn't do the experiment. Okay, guys, if you really have trouble with this more exact lifespan or longevity, if you have still questions about this, let's meet for extra help next week because I have to teach you about lipids and this is going to take us the rest of the period, okay? And it goes in the quiz tomorrow, so we want to learn it. So I'm going to go back and go to the stream and find our PowerPoint of macromolecules. And then in macromolecules, we're going to go to learn about lipids. And I put the video of this, if you want to review it later on, you can. So for lipids, we are going to start by learning about, so first of all, the definition of lipids, right? So lipids are a group of molecules that are grouped different than all the other macromolecules. So all the other macromolecules have a polymer, a monomer that repeats. This group is very strange because they are basically grouping different molecules that are not similar to each other. They have more than one kind of lipid. And the only thing they have in common is that they have very little affinity to water, or in other words, they have very low polarity. Okay, and it does say little or no, because some of them have a little affinity to water. So it's a very strange group, disparate group of molecules, okay? And the first kind of lipid that we are going to learn is called fats, okay? Fats are their own molecules. So there's three main lipids that we're going to learn. Fats are one of them, and they are constituted by glycerol and a fatty acid. So what are they made of? So a glycerol, which is this alcohol, is called a glycerol, okay, a 3-carbon alcohol called glycerol, and it joins three different chains of fatty acids. This is one example of them. It's a carboxylic acid, okay, called palmitic acid, but there can be many of them. So when the fatty acid joins the glycerol, this is a dehydration reaction. And this dehydration reaction that will make you the fat will always form this bond called an ester linkage. So you have the 3-carbon alcohol called glycerol, the 3 different fatty acids, and the carboxylic group of the fatty acids will donate the hydroxyl. The hydrogen will come from the alcohol, and that water will leave by dehydration, and that will form a bond called ester linkage. So it's similar to how you saw glycosidic linkage, but this one is called ester linkage, and it's what makes you a fat molecule, also known as 3-acylglycerol, or you may have heard of them as triglycerides, when people say, oh, I have very high triglycerides. Okay, so before I go on, I would love to hear if there's any questions about that. You will get extra help on lipids, I guess, next week. Yeah. Or you can ask me questions at the beginning of class tomorrow. Okay? So fat is its own molecule. It's its own type of molecule, yeah. So any more questions about the ester linkage that forms to make the fat, to make the glycerol join the carbohydrates, the carboxylic acid? Aren't all molecules their own molecules though? What I mean is this. Within the macromolecule called lipids, there is groups of lipids. One group of lipids is called the fats. Another group you're going to learn after this, and there's another group. So that's what I meant. So lipids are this. They are all made of a glycerol that joins 3 carboxylic acids, and the carboxylic acids that make up the lipid will vary, but it's always a glycerol with an ester bond. Okay? So I'll continue. And yeah, but guys, so in terms of the whole discussion we had with the quiz, which obviously there's a lot more confusion, right? Because I see there's a lot of questions. I asked you questions when the class starts and nobody says anything. So I think we need to do a bit of a better job at that point. When the class starts and I say, hey, questions, if you've been doing your work, writing your notes, you probably have written things that you don't know. Unless you have not been doing that at all. Not copying the notes, not writing anything, and you just show up every day and you've never been reviewing anything. And then you won't have questions because you are facing the questions the first time when I give you the quiz. But if you are, and if it's because people are feeling shy, I'm going to encourage you to ask questions, because the more questions you ask, the more you'll learn, the more we'll be able to help you. Or I'm going to encourage you to attend extra help also, because even though, yeah, today when we did extra help, we hadn't learned lipids, I also didn't have a lot of people from this class in extra help. And last week, I didn't have a lot of people for extra help either. So, it's important that you guys ask the questions when the questions are being offered so that we have a chance. Okay, just forget until we get there. Yeah, okay, well, make note, write it down. Could you post the answers for the textbook questions so we can have extra review? Which textbook questions? You mean the ones at the end of the chapters in PowerPoint? Oh, I don't have that. I don't have that. I only have the whatever I post I have. I can check if it's available, but I mean that PDF of that textbook is not mine. Somebody else gave it to me so you guys could study. But what we can do, this is what we can do, Jusra. If there's questions in there that you're like, oh, I don't know if it's right, bring them to class. Come the next day and say, hey, Miss, I didn't know what this was. And if it's not too involved, I'll answer it for you. Okay? So when you say lipids are hydrophobic, yeah, all lipids are hydrophobic, or variation of hydrophobicity, variation. Okay, let's keep on going. So the ester linkage makes this molecule called triacylglycerol, which is the molecule that makes up a fat. Now, there's 2 kinds of fats. You can have what's known as saturated fatty acids to make up a fat, or unsaturated fatty acids. And when we're saying saturated or unsaturated, what we're referring to is this, right? In there, you don't have any double bonds, or you have double bonds. So what do you know? Why do you think it's called saturated, unsaturated? What is it saturated of? Grace, it couldn't be atoms. That makes no sense. Of course, if it's a molecule, it's got to be made of atoms. They both have atoms, so that makes no sense. There's a lot of bonds. A lot of bonds with what? Because they both have bonds. So when it's double bonds, it's called unsaturated. So what is it unsaturated? What doesn't it have? Hydrogens. It doesn't have hydrogen. So when it's saturated, it has lots of hydrogens, and so it's saturated with hydrogen. When it's unsaturated, it doesn't have as many hydrogens, so it ends up making double bonds. Oh, no worries, Risha. As long as you ask questions when you get lost, if you by a moment have to leave, ask questions. John said, but what? There below doesn't look like it's missing. Yeah, I know why you're saying that. This is a simplification, the diagram, okay? Every corner is a carbon. It's not even showing you the hydrogens, but in this one, it's showing you a double bond. So even though it doesn't show you the hydrogens, it's showing you the double bond. So that's how you know this is an unsaturated, and this is saturated. It means it has full hydrogen everywhere. And this drawing shows you, and this drawing shows you the double bond and the less hydrogens in here. Okay? Now, what do you notice about saturated and unsaturated? Look at the picture. Look at the picture. What do you notice about saturated and unsaturated? Unsaturated is bent slightly. You're right, Oiseme. So unsaturated is bent slightly, not saturated. Unsaturated because the double bond, remember, is not flexible, right? It's fixed. So if it happens there, then it's going to make them stick out like that, right? So the double bonds, which are the unsaturated fats, they don't have as many hydrogens, so they are double bonding. Those have like a kink. What else do you notice about them? What is different between them? Look at the pictures. What did I have in the pictures? Saturated is solid, unsaturated is liquid. Why? It's funny, right? Unsaturated is always liquid, saturated is solid. There must be a relationship. Why? Yeah, well, the hydrogens are what makes the saturated molecule saturated. Well, why is the saturated molecule solid? Because the double bonds make harder to break apart. Why would that make it a liquid? Breaking apart the bonds will break the material. It doesn't mean it'll change the state. Bonds are stronger. The stronger bond doesn't change the state of a molecule. Something to do with the bend shape, probably. What do you think it is? Think of everything you know about solids and liquids, and think what you see in the picture. And it makes sense. Isomers? How is that to do with solids and liquids? Why would it be something that has that double bond and the kink always liquid, while something that doesn't have the double bond is always solid? And think of what you know about solids and liquids. What do I know about solids and liquids? What's the difference? Exactly, Olivia, good job. The unsaturated have more space between the molecules. So yeah, because if you have that bent shape, you have a lot more bulk. If you have a lot more bulk, you have more space between molecules, hence your liquid. If you have more compact spaces with single bonds, then you compact, then it's solid. Yeah. Okay? So that's why the saturated fats tend to be solid and the unsaturated fats tend to be liquid. Now, there's something else that is interesting. Here there's butter, here there's oil. What else do you know about butter and oil? What do you notice about that you know because you know from eating? Butter and oil are the examples. Another saturated fat that exists is lard, which is made from pig's fat. And unsaturated fats examples can be olive oil, sunflower oil. So what do you notice is difference between the liquid and the solid also? Aside from the fact that one is solid, one is liquid because of the... No, that's not necessarily true. That's not always true. But think of where butter or lard comes from and where olive oil and sunflower oil, exactly. So saturated fats tend to be animal source and unsaturated fat tend to be plant source. And in general, animal fat is less healthy for us than plant fat. Okay? So in general, it's much better for us to consume fat that is like olive oil, sunflower oil, than consuming butter or lard. It's bad for us because we start building up, the fat builds up in our arteries and it makes kind of a clog, and it causes heart disease, right? So animal fat is not as good for us than plant fat. Now, doctors, when they discover this, especially because in North America, in the US especially, this used to happen. People would have for breakfast butter or bacon almost every day. And especially men in the 1950s and 40s, they started dying of heart attacks very young in age. And doctors discovered that it was all the animal fat. So they figured, but if you don't eat animal fat, it's healthier for you. So if you had to figure out how does a person that loves butter get to have butter, but it is not animal source, what would you do? What would you invent? You know they love a plant-based butter. How would you do that? Like a vegan butter, some kind. What would you do for that? How would you make that? Using almond milk? You still haven't answered my question. How do you make it solid? You have olive oil or sunflower oil or all these oils. I want to now make them into a solid thing. Keep it chill? Really? Have you ever put olive oil in the fridge? It doesn't become butter. No, think of the chemistry, people. How do you make it into a butter or solid? What do you need to do? Thank you, Usama. You remove the double bond. So what do you have to add? Exactly, Natasha. You add hydrogen. You hydrogenate it, and then the oil will become butter. Good job. So that's called margarine. That's how they make margarine. Margarine is made with the oil and they added hydrogen. But you thought it was fantastic, right? Then what ends up happening is not so good. So yeah, they add hydrogen. They call it hydrogenation. It's a reaction called hydrogenation. They pump hydrogen at high pressure and it starts making solid. Yeah, but this is what ends up occurring. If you notice about this fat, the double bond, going back to our isomer idea, this is a cis double bond, right? You see, it's a cis double bond. When you hydrogenate oil and you make margarine, as a secondary effect, you sometimes end up with a little bit of oil that is trans fat. Because it's artificially made, you've actually pumped all the hydrogen in there. Some of the molecules will end up kind of rearranging and making a double bond that is trans. Some of them will become solid, but then some of them will make trans fats. Now, you've heard all your life, trans fat, no trans fat, trans fat is bad for you. Yeah, you make it saturated, but sometimes there'll be a few left over that end up being trans fat. Yeah, there's cis fats, which are the normal oils, and trans fats, they are a consequence of rehydrogenation. So if you remove the double oil, becomes butter, Sebastian, or margarine. Margarine, not butter, because it's artificial. So, why do you think that trans fat is bad? You've heard all your life it's bad. Why do you think it would be odd for our body to deal with the trans fat? Why would that be bad? It's not natural, and then what will happen to your body with it? Yeah, you don't have the enzymes to break it down. So it's not even harder. You cannot break it down at all. So it ends up building up in your arteries and clogging your arteries, and it actually makes it even worse than if you had eaten butter in the first place. Exactly. So that is the problem with hydrogenation. So what occurs, and this is written all here, we discussed it, but it's written. So most animal fats are saturated, which are unhealthy for us. Sometimes they created hydrogenated vegetable oils, which made butter, but it creates trans double bonds, and that is extremely dangerous for us, and it causes heart disease. So that is an important aspect of how artificially created margarine can actually be worse for you. No, you cannot get it out. They start building in your arteries and your vessels, and that's why people. Exactly, Shawnees. The goal was to make us healthy, and it ended up being worse. So once again, the only solution to be healthy is moderation. Don't eat too much butter. Don't eat too much cheese. Do exercise and eat veggies and fruits, and then you'll be fine. Well, we can unclog the fats if those fats were natural. Okay. Don't a lot of people eat margarine? Well, now they are making margarine in different ways, but yeah, I think that the best choice is to simply not eat too much butter and just eat olive oil and healthy oils. Yeah, butter is better. Natural is always better. Yeah, sadly. Well, my life of eating butter is flashing before my eyes. No, that doesn't mean you cannot eat butter. I love butter. I just don't eat too much. Don't eat too much cheese or too much butter. Green butter, corn. Yeah, I'm making that tonight for dinner with veggie burgers. Veggie burgers, though. Okay. So let's not digress because we still have other lipids to learn. Phospholipids. So phospholipids are another molecule that is a lipid. Okay, and this is different than fats. This is like this. It's made up with one phosphate group at the top and two carboxylic acids at the bottom. So this molecule, as you can imagine, is quite polar in this part, but it's non-polar in this part. So this is, for instance, an example of a lipid that is partially polar. It's a little bit soluble in water, but even though this top part of the molecule is polar, the bottom isn't. So if you put it in water, this is what it will do. It will have one section that is in the water and this section that is hidden from the water. Now, have you seen this before? What do you think that looks like? Well done. Yeah, that is like the cell membrane of most living things. The cell membrane of most living things is made up of phospholipids in a bilayer, 2 layers. And the part that is exposed here in this example is the part that is in water. Now, our cells are not in water. What is the water part? What would the water be? Exactly, the blood. So the part that is connected to the blood will simply be the polar section of the molecule, and there will be easy connection, and then the non-polar section of the molecule is hidden in the middle like a sandwich. Now, say that I grabbed those phospholipids and I put them in a glass with oil, like this. I said to you, put them in here, in a glass with oil. What do you think it will look like? What would happen? They would be separate. Well, look at this, right? You have water here, water here. This is the hydrophobic section. This is the hydrophilic section in the middle, and then the other side hydrophobic section. If I put them in oil instead of water, what do you think they would do? They would separate. Take your time. Hydrophilic, it's in the middle. It's the opposite, exactly. So they would be, instead of like this, they would have the hydrophobic section in the oil and the hydrophilic section in the middle. It's like if you made a sandwich, but now the sandwich is cheese, bread, cheese. Does that make sense? Oh, Nicole, you're an artist. That is fantastic. Good job. Yeah, that's it. Sebe, I think you should look at the drawing that Nicole did, because it's just spot on. You are in the oil, so the tails that are hydrophobic are exposed to the oil, and the hydrophilic head of the molecules inside. Cool. Okay. So that is what occurs with phospholipids, they matter a lot because it's what makes up our membranes. And the last type of lipid that we will mention, and by the way, this PowerPoint doesn't include a fourth kind of lipid that exists called waxes. So I'll just mention it to you. So waxes are, for instance, you know how your apple is very shiny on the outside, or a plum? That's made up with a wax, so that the rain will slide out and not rot the waxes. Yeah, but we won't learn the molecules. Candles also are made up of waxes, yeah. Okay, so steroids is the third lipid that we will learn. An example of a steroid is cholesterol, testosterone, most hormones are steroids. And basically the only thing you have to know about them is that they are made up of fused rings, and they look sort of like that. So this is cholesterol, but you can look at steroids of any kind. They usually are, hormones are usually steroids. So you don't need to know waxes because it's not even in the PowerPoint. Just I'm mentioning it, I'm just saying it's another kind of lipid. So it suffices to know that waxes can be a lipid. That's all. But yeah, we're not spending more than that. Cool. So yeah, so that's basically the end of our class. We only have like 10-15 minutes. I had another section, another practice I wanted us to do in Explore Learning. But before we go there, I would like to hear questions, parts that you want me to go over, so that we don't leave this until we are good, that we understand. So unsaturated is better because it doesn't, so the whole unsaturated and saturated, it has many layers. So number one, saturated fats tend to be solid, unsaturated fats tend to be liquid. Saturated fats that are solid with natural conditions, they come from animal sources, while unsaturated fats that are liquid in natural conditions tend to be from plant sources. In natural conditions, saturated fats tend to be a bit less healthy for us because the solid materials from animal source tend to give us heart disease. Then, doctors try to make artificially solid saturated fats, so they took natural oils and pumped them with hydrogen to make margarine. The problem with that is that then they created artificially oils that were, as a byproduct, trans fat, and those are actually worse for you. So it's kind of step by step. So yeah, write it down when you clean your notes, write it down step by step so you don't get confused, because it's a lot to it. But shouldn't it be in a liquid? Yeah, it would. It would be in a liquid state. The trans fat would be a liquid. It's like there's a difference in bonds. Yeah, the cis and trans. So can we process saturated fats? Yeah, we can. You can eat butter. Yeah, you can. But too much of it makes too many fats in your blood, which also makes plaque in your arteries. That's why people get cholesterol problems and heart disease. And for the olive oil, no, the olive oil is fine for us. The cis fats is fine for us. I mean, too much has too many calories, but it's fine. I'm going to ask you a difficult question, guys. What do you think has more calories, more energy? A teaspoon of a monosaccharide, a teaspoon of a polysaccharide, or a teaspoon of oil? Oil, Sunny says oil, polysaccharide says Nicole. Kiva just wrote, oh, oil. Natasha says, we're supposed to eat these things, for example, are they necessities for human body, or we eat just for fun? Okay, well, let me address the energy question and then I'll explain. Oil, polysaccharide. So, okay, so let me go back to the PowerPoint for a minute and you'll see the answer. Okay, let's go back to carbohydrates for a sec. So look at the carbohydrates, right? When I say polysaccharide, we know that a polysaccharide is made up like this, of very long chains of glucose, right? Look at this, how much it has, right? And this is just the one glucose, right? So a monosaccharide is a teaspoon of these dude, a teaspoon of those guys, these things, or a teaspoon of oil, which would be a teaspoon of this one, but with double bonded. So if you think about it, what do you think will have more energy and why? Monosaccharide is one, polysaccharide is the long chain. One glucose versus a long chain of glucose that curls like this. Oh, which specifically, any. So a teaspoon of a monosaccharide, a teaspoon of a polysaccharide, and a teaspoon of any oil. So Nicole is saying it's fat. Why did you think it's fat? When you look at the pictures I showed you, why did you think it was fat? Notice I am saying a teaspoon, a teaspoon, a teaspoon. So this is, by the way, an experiment. What's the independent variable here? Yusra, the independent variable is not a teaspoon. The teaspoon is a constant, man. The molecule type is the independent. And what's the dependent? The energy content. Okay. Now, we are saying that the fats have, well, majority of people chose fats as the most energy. Why are you saying that? Why do you think the fat has more energy? Let's go back to the picture. Oh, and then you Google that. That's not the way to answer the question. That's cheating. Come back here. Look at the picture. What do you notice about this picture versus the sugar picture? So look at this picture of a sugar. Look at this picture of the lipid. What do you notice? Exactly. It will take, in the same volume, a lot more condensed bonds, because you don't have these big rings, you don't have these big coils. So it takes a lot less space for the same amount of bonds, and bonds are energy. So then it's a lot more energy in something smaller, right? The molecules are the explanation for everything now. Does it make sense that we store our energy as fat for long term? What would happen if we store our energy for long term as carbohydrates? What would happen? They would up quickly, you're right, but also what would we look like? Yes, Shawnies. And what would you look like if your energy was now stored as a carbohydrate? What would you look like? It was funny, bread. It would take a lot more space, right? And taking a lot of space for human beings is a problem. Why is that a problem? Why can we not afford to take up so much space? While plants don't have to worry about this, but we have to worry about not taking too much space, because what do we need not to take space for? Why is that a problem? It can be stuck on populated, but now think about animals, for instance. What do they need to do? Like, they have to move around to hunt and eat food, right? So to be efficient and store energy as fat allows you to have an efficient little source of energy for when things are tough. Exactly. So I think, see the beauty of this, how chemistry and biology are like twins. They all connect, the function and the form are connected. Now, let's answer the interesting question that I think it was Olivia that was asking. Which foods are you supposed to eat and which foods are for fun? Which is a very important question. Okay, so of course, according to dietary sources, you do need to consume uh, sugars for energy, immediate energy. You cannot exist. I ate too much cheese that shines. Okay, so dairy, there's an energy guide of how much dairy you're supposed to consume. You're not supposed to consume too much dairy.
Is it small
You're not supposed to consume too much fat, but you need to consume fat, females especially, all the hormones and stuff. You need to consume dairy a little bit, especially for young people like you guys, because you're still growing, you're still making your bones strong, so you do need to consume dairy. And you need to consume some proteins because you're still growing, believe it or not. And it's very important you consume veggies and fruit because it helps us fight diseases and stuff. And, keep active, especially now in quarantine. Try to exercise. So if we do all those things, we're doing it right. Doesn't matter if you love cheese and you eat cheese. As long as you balance it, it's fine. Okay? Does that make sense? Does that help? Too much fat is not great. Can you explain how margarine is made again? So I'll show you, because the period is about to end, I'll show you quickly. So what they do is they take the saturated fat, like this guy that we see in front of you, and the unsaturated fat, like this one that you see in front of you that has double bonds, and it would have probably a lot more double bonds, and they pump hydrogen into it. So that forces it to become like this. And that makes it a solid because it stacks up. But when you do that, as a byproduct, a little bit of the oil converts into a trans fat, and that one is the one that catches you. That's the one that is bad for you. Hydrogenation.
Wait, miss.
Mm-hmm.
Wait, so I didn't really hear it because kind of but you said they basically take the unsaturated fat or the saturated one? The unsaturated one, right?
The unsaturated one, they pump hydrogen,
in it.
and then that makes it solid, makes it hydrogenated and solid, and then a little bit of the oil converts into trans fat, and that's the one that is bad for you.
Also, a little bit of the oil converts into trans fat, and that's the one that's bad for you, right?
Yeah, exactly.
Oh, the oil from where?
Sorry?
Like the oil from where?
Well, what happens is that you had oil to make it into the margarine, right? So the original oil, some of it will break and rearrange, and when it rearranges, it won't make a cis again. It'll make a trans.
Oh, okay. I see. All right, thank you.
No worries. We need to know about steroids again. Not much, really. Steroids are a few small ring molecules. They tend to be hormones. Examples are cholesterol, testosterone, estrogen. That's all, really. Not much more than that. Can we wait? Can you please go over phospholipids again? I forgot I asked. No worries. So phospholipids are another kind of lipid. It's made up with a hydrophilic head made of phosphate and a chain of fatty acids, and they make up the membrane of the cell membranes. Yeah. So guys, review your stuff for tomorrow. I put in Explore Learning a dehydration gizmo that I think you guys may find useful. So when you're reviewing for your quiz, do that gizmo and Explore Learning, because why not? And go through your notes, and then when we start class tomorrow, I'm giving myself a few minutes to answer questions. So your quiz won't start right away. You have about 10 minutes to answer questions, to ask questions, and then we'll do the quiz, and then after that, we'll do the lab, which is exciting, whatever that ends up looking like. Okay? So make sure you bring your stuff for your lab. Put it in the kitchen, and then we'll go and do it. What differs between trans and cis if the double bonds are still in the carbon? Yeah, but the chain is either like this or like this. It's either the same side or different side. And you cannot break the ones that are the other side. Yeah, it's the shape. It's just the shape, nothing else. They're isomers, right? So it's just the shape. Same molecule, same everything, just the shape. Okay, so I have a recording of the lesson and I have the PowerPoint in the Google Classroom. So go and have a look and review that, and any more questions you have, you can bring tomorrow. Yeah, well, I'll tell you tomorrow anyways. You'll go do it, either take a picture or film it, and then we'll discuss a few minutes after. Okay, we're good? Okay, guys, so I'm going to say goodbye.